Hi there, welcome back. We're going to be working on Bio 232 lectures. We're now going to go back to vessels. We did structure a couple lectures ago, and then we stopped and worked on structure and function of the heart. And now we're back to looking at the peripheral vessels, and we want to look at what things affect them, including hormones, blood pressure, how they affect blood pressure, so on and so forth. So that's where we're going to go now. So we're going to go back to our blood vessel notes on page 7, and we had stopped right at the top of the page last time. We're going to pick up where we left off. Now this lecture, I'm going to go over a few pages of these notes. The next lecture, I'm going to use the notes that I made for hormonal control of blood vessels. That's on your Moodle page. So on this week's slot, you'll see it there. So you could bring that up for next time, but we won't need it right now. We may review a little bit of the concepts we talked about today in the next lecture also. So really what I want to do is define some terms and kind of start to understand how circulation affects blood pressure and other parts of other aspects of our body. So first off, we need to define a couple terms. First, blood flow. So blood flow is just movement of blood through a closed system of vessels. All right, so anytime we have any fluid moving through a closed system, we can watch its flow, right, the movement of it. And this flow occurs, it is due to different pressures. So blood flow is due to different pressures in various parts of the system. And we always are moving from a higher to lower pressure. And we always see this, right? We always go from high concentration to low concentration, things like that. So where there's a higher pressure, blood will want to move from that high pressure to an area of lower pressure. Okay, so that happens most of the times. Now, under resting conditions, blood flow is generally equal to cardiac output. Remember, we had our cardiac output equation. But we're often not in resting conditions, and so these two things will not be the same. All right, now we need to define blood pressure. We've already been using blood pressure, and most of you kind of understand blood pressure, but I'm more kind of trying to be specific. What does blood pressure really refer to? And so blood pressure, especially when we're talking about in the arteries, the systemic arterial blood pressure, is the pressure exerted by the blood onto those walls of those vessels. Now, generally, we refer to the arterial pressure, right? When we talk about 120 over 80, we're looking at the blood pressure in the artery, and often the brachial artery if we are measuring blood pressure on our arm. So blood pressure is pressure exerted by the blood onto the walls of any blood vessel. But remember, we can measure blood pressure other places. We can look at pulmonary blood pressure, the pulmonary system. We could look at blood pressure coming out of a capillary bed in the venous system. And it's measured in millimeters of mercury. And this is just because back in the old days, they had this mercury in a column, and if you applied pressure to it, the mercury would rise up a certain number of millimeters, and whatever the pressure is that you applied, we would say, oh, it went up 7 millimeters of mercury. So that's how we still measure it in terms of millimeters of mercury, but we, of course, don't use mercury in that pressure system in our modern-day blood pressure measuring devices. Now, the pressure that was in our closed system causes the blood flow to happen. But things can impede blood flow, and we call those things resistance, specifically peripheral resistance, anything out in the periphery of our body, the vessels out in our body that are going to slow down blood. So this impedes blood flow by the force of friction between blood and the blood vessels. One way to think about this is if you have a blood vessel in cross-section like this, and blood is moving through, and you have blood cells, and you have liquid in it, there are going to be times where the liquid and the blood vessels are going to rub up against the wall. And when they bounce and hit this wall, they're going to cause friction. And that friction is going to slow down the system, right? And luckily, because it's a liquid system, the blood flows pretty well, but there's still small amounts of friction as the plasma and as the blood cells rub up against the walls of the vessel. Okay, so where do we see this resistance? There are really three things that kind of alter resistance. All right, and they're right here, these three sources. So the first is blood viscosity. This is how thick the fluid is, okay? This is the stickiness or thickness of the fluid. The thicker the fluid, the slower it moves, right? Think of molasses or syrup as opposed to water, right? The thicker the fluid, the more resistance. The second is vessel length. This means if we increase the length of our vessels, there's more walls for the blood to rub up against and create more friction against. So when we increase our body size, we increase our vessel length. So if you start to gain some weight and put on extra weight, you need more blood vessels in that tissue to supply it, and therefore our vessel length increases and our peripheral resistance would increase. But generally, these first two are mostly constant. In a normal individual, our blood viscosity doesn't change very much, and our vessel length doesn't change very much. All right, the thing that does change a lot, and we'll put it in a different color just to kind of really highlight it, are changes in vessel diameter. And this is because the arterioles can change their diameter by vasoconstricting, vasodilating, and they can do it minute by minute, all right? And I don't know why it's giving you a spade there. What it should be doing is this is a delta. And delta means change in pressure, okay? So if we have a change in pressure, all right? And so what we're saying here in this little equation is blood flow, which remember, under normal conditions, hold on, oh boy, you can never mess too much with this. All right, what did I do? I lost a whole page there. Okay, still lost it. All right, I'm going to pause. Okay, I'm back. This thing messes up sometimes and makes a lot of weird things happen. I love my OneNote program, but once in a while things happen that are strange. Okay, so I had changed these spades into little deltas, right? These little deltas. Oh my goodness. Delta. Okay, and over here, delta. All right. Now, this third source of resistance, this is the one that is most important because we can do this due to the smooth muscle in the arterioles can contract and vasoconstrict, or can dilate and or can relax and vasodilate. So blood flow, which is the same as saying cardiac output in a resting individual, right? We talked about cardiac output is equal to blood flow under a lot of conditions, all right? It is proportional to pressure changes, meaning that if blood pressure goes up, cardiac output goes up, blood flow goes up. That makes sense, right? If you increase the pressure of something, more blood will flow through and more cardiac output, more blood per minute, right? If you increase the pressure on the hose, the garden hose outside your house, more water goes through per minute, all right? It's inversely proportional to resistance, okay? So what we mean by that is that the more resistance you have, the lower the blood flow. Right? Another way you can write this equation here is you can say cardiac output or blood flow is pressure change times 1 over resistance, right? That is the same as that. So when we do the 1 over, it means that if this number goes up, cardiac output will go down. If cardiac output goes up, if this number is getting higher, then that must mean that this number is lower. Right? So they're inversely proportional to each other. Now, one way that we can change resistance is changing the size of the vessel. A larger diameter compared to a smaller diameter, there's more volume and less surface area, so there's less friction that will happen. Most of the blood is going to flow in the center here. It's not going to hit the walls for friction. But here, there's a lot more surface area as compared to the amount of volume, so there's more friction relatively. So an increase in lumen size, this is the lumen here, leads to more blood flow and less resistance. And again, you can think of that's kind of intuitive. If we have a bigger pipe that water's going through, it will go through at you can have more blood go through per minute, and the resistance is lower. But if we have a really small pipe, we can only get so much blood in through that pipe per minute at a time. Okay, so that's the first thing. Second thing, now let's define some pressures a little bit. Okay, so we have systolic arterial blood pressure, right? This is the top number of our, if we say, if we were going to say an average blood pressure is 120/80, systolic is that, diastolic is this. All right, so what does systolic mean? Systolic is when the blood flows from the left ventricle to the aorta. The aortic pressure is at its highest. And if you remember, when we looked at our cardiac cycle diagram, the peak of the aortic pressure was at 120 millimeters of mercury. This will push the blood all the way through the body, down to the tissues, and eventually back to the right ventricle. Now, of course, it needs some help, especially in the venous end, but that pressure is very high. Now, diastolic pressure is when the pressure is at its lowest. So after the heart's contracted, the heart's in its, the ventricles are relaxing and repolarizing. Diastolic pressure is in a resting heart, around 70 to 80 beats, 70 to 80 millimeters of mercury. Okay. Now, the mean arterial pressure is to do this calculation to find what the mean is, or the average pressure. I don't use that very much in our stuff, so I'm not even going to talk about it. Pulse pressure is where you subtract the two, and you get the difference, okay? And so you'll sometimes see that term used as well. But again, I don't use it too much. I usually use systolic blood pressure and diastolic blood pressure. Systolic and diastolic, and that's it. All right, now we want to talk about factors that affect blood pressure. So what I have here is a little diagram, and I like this little diagram because we can write in some things like blood pressure, and we can write three things that affect it. Now, we just talked about peripheral resistance, right? We just talked about blood volume. And we were talking about cardiac output a minute ago, and how cardiac output and blood pressure are related. So cardiac output, remember, is SV times, or actually we can do it this way, HR times SV. Okay? Now, the other thing to think about here is, how are they related to each other? Do they go up or down? So if we increase one of these, will the other one increase or not? And that's why the arrows are pointing from one thing to another. So we're basically saying, how does this affect blood pressure? So what we can do is we can start to draw in some arrows that will tell us some things about this. So if we have an increase in any of those three things, cardiac output, heart rate, or stroke volume, we will increase blood pressure. Right? So think about that. If our heart rate goes up, our blood pressure goes up because more blood goes through, is being pumped per minute. If our stroke volume goes up, it's because more blood is being pumped out at each pump, each contraction per time. And so more blood is in our vessels, and it increases pressure. And then the combination of those two, if cardiac output is higher because of one or both of these, then blood pressure will be higher. Okay? So remember, cardiac output is the amount of blood ejected by the left ventricle in the aorta each minute. Right? And you can also say right ventricle. It doesn't matter. We're using left most of the time. And so that's the equation. And so if cardiac output goes up, blood pressure goes up. Right? So, and this is, I don't know why, but that arrow worked, but this one didn't. So if cardiac output goes up, blood pressure goes up. That's what we just said. Now let's look at the second one, peripheral resistance. Peripheral resistance is impedance of the blood flow by a vessel wall, and this is due to friction, right? We just talked about this. The function of the arterioles is to control peripheral resistance and in turn blood pressure by changing diameter. We just talked about that. If we vasodilate, okay, that means there's more space for the blood to flow. There's a bigger lumen, and that means that peripheral resistance will go down, right? If that goes down, blood pressure will go down. So let's look at this. Let's say we make the lumen wider, peripheral resistance goes down, blood pressure goes down, okay? Now, let's go back here for a second. What if cardiac output goes down? What will happen to blood pressure? Cardiac output goes down, blood pressure will go down. What if the heart rate goes down? Blood pressure will go down. What if stroke volume goes down? Blood pressure will go down. Okay, now let's go back to peripheral resistance. What if we vasoconstrict and make a very small lumen and the blood that's being forced through is rubbing up against the walls a lot? That means that resistance goes up. If resistance goes up, guess what? Let's make a better looking arrow. If resistance goes up, blood pressure goes up. Okay? You getting the hang of it? Pretty straightforward. So the arterioles control peripheral resistance and in turn blood pressure by changing diameter. The last one is blood volume. This is approximately, that's supposed to be an approximation, not 0.5 liters, just 5 liters. This is supposed to say approximately equal to 5 liters. It's usually constant. But let's say we were bleeding a lot. Say we had a very bad bleed of some sort and our blood volume went down. Guess what? Our blood pressure would go down. Let's say we pump someone full of a lot of fluids for some reason and their blood pressure goes, or blood volume goes up. Guess what it'll do? Their blood pressure will go up, right? So notice these arrows work in all three of these scenarios. If one of those goes up, it affects blood pressure upward. If one of those goes down, it affects blood pressure downward. Okay? Now, keep that in mind. Blood volume. So again, these 8s are here. I'm sorry, these silly 8s. They're supposed to be up arrows. If you didn't notice them, I would have fixed them. Increase in salts would increase blood volume because water stays with the salt. This, here, let's put a bracket around these. This, salts leads to fluid retention. Fluid loss would mean low blood pressure. Extreme dehydration would be low blood pressure. Actually, let's do this because I don't really like the way this is set up. If we had, blood pressure would go down with hemorrhage. It would go down with extreme dehydration. It would go up with salt and fluid retention. So I'm going to put a little note here. That would be increase in blood pressure here. And if we lost a lot of fluid, we would have a decrease in blood pressure. Okay? So hemorrhage would lower blood pressure, extreme dehydration would lower blood pressure, increased salts and therefore fluid retention would increase blood pressure, and if we had a lot of fluid loss in some reason, we would decrease blood pressure. Okay. Now, we talked a little bit about the regulation of blood pressure, nervous system controls for the heart, but we haven't really talked about it for the vasomotor center, for the vessels. So that's what I want to talk about for a few more minutes here before we end this lecture. So we already said in our last lecture that if we increase our heart rate or the force of contraction, cardiac output goes up. And now we know that cardiac output is directly related to blood pressure. So if cardiac output goes up, blood pressure goes up. All right. So, and we know that at the heart, we can have sympathetic controls on the heart that does that. That's our CAC. But also, we can have a sympathetic response to the vascular system. So when we have a sympathetic response, we can go to the vasomotor center in the medulla. And these control the efferent fibers that go out to vascular smooth muscle, the smooth muscle in the tunica media of the vessels. And it's a sympathetic-only system, okay? Sympathetic-only system. The nuclei in the medulla only control the diameter of blood vessels, and all they do is they constrict them. Now, normally, we have slightly constricted blood vessels all the time. So, generally speaking, the vasomotor center is sending impulses to vessels, keeping them in moderate vasoconstriction. And this is called vasomotor tone, or sometimes just called vasotone. So I'll put that in because you sometimes see it as both. Vasotone, kind of all together. So the more impulses that we have at the vasomotor sense, the more constriction, and the less impulses, the less constriction. And that is indirectly vasodilation. This is why we don't need a parasympathetic, because all we have to do is just stop sending sympathetic messages, and the blood vessel will vasodilate. Okay? So if we didn't have any sympathetic impulses going to them, they dilate fully. Does that make sense? They just dilate completely fully. Okay, so we call these vasomotor fibers. These are the efferent fibers. They're sympathetic. They go to the smooth muscle of the arterioles, and they release norepinephrine as a neurotransmitter, and they cause vasoconstriction. Now, we also have those baroreceptor-initiated reflexes, and we talked about these already a little bit. The carotid sinus and the aortic arch we talked about last time, I believe. Blood pressure increase will stimulate these receptors, okay? And so if in our carotid arteries or in our aorta we notice a lot of blood pressure, that will cause the impulses of the vasomotor center to inhibit their activity. So we want, if we notice there's a lot of blood pressure in our aorta and our carotid arteries that go up to our brain, that worries us that our blood pressure is getting too high. So what it does is it tells the vasomotor center to inhibit the activity and allow vasodilation of vessels, both arteries and veins, but mostly arteries, and it results in lower blood pressure in our periphery, right? Because what it'll do, I'm going to go back up here, is it will make the lumen size bigger, which will lower peripheral resistance by vasodilating, and that will lower blood pressure, okay? Now, it'll also, and we talked about this before, the CIC will get stimulated and we'll have a parasympathetic response, and that will cause cardiac output to go down, right? And we know that lower cardiac output, let's go back to this, lower cardiac output here results in, lower cardiac output right there results in lower blood pressure. All right, so that helps as well. All right, now this next page I don't find as useful, but if it does for you, great. It's a way to kind of just show all these different things happening at the same time. And you can put little arrows in this top one where you can increase vasomotor activity and then increase the impulses and decrease or increase the vessel size. I'm not going to do that. I don't think that for a lot of people it gets very confusing. I think what I said is better. Now, these are the baroreceptors, and the same thing. This is detecting what the barodetectors notice due to high or low blood pressure and how they react. Now, again, this is probably more complicated than you need to know, but if this helps and you can put everything together, great. All right. Now, let's see. I may cover this in the next lecture again, but it's fine for me to go over it a tiny bit right now. There are chemoreceptor-initiated reflexes, and these are in the carotid and aortic bodies as well. And these are where these parts of our vessel system, underneath from our heart to our brain area, they're very sensitive to certain things like arterial oxygen, carbon dioxide, and pH. So what it looks for is it says, do we have too little oxygen? Or do we have too much acid or too much carbon dioxide? And if so, we want to increase blood pressure because we want blood to go through the system faster and get more of the deoxygenated blood in the venous system back to the heart so we can pump it to the lungs and get more oxygen. So if we notice any of those things, The vasomotor center will increase its sympathetic stimulation. This will enhance vasoconstriction, and that will enhance blood vessel pressure, which causes quicker venous return. Okay? So this is an important thing that we see sometimes. Now, your brain can also cause changes. So if you have intense anger, that can cause sympathetic stimulation. That'll vasoconstrict and increase blood pressure. Think of someone who's so mad, they're getting so angry, their face starts getting all red, their blood pressure is increasing. You can just see their blood pressure increasing. Now, what if you're really emotionally upset, maybe depressed long term, for example? This would actually do the opposite. It could lower sympathetic stimulation. And in some cases, you can have like a temporary emotionally upset where you're so upset you vasodilate too quickly and you faint, right? So that does sometimes happen, and which is what this little part is saying right here. We get vasodilation, and our blood pressure drops so much that we go into like a little temporary shock and we faint. Your hypothalamus, of course, can help with this too, because it is doing so many different things for our homeostasis that's almost just, of course it is. So I'm not even going to mention anymore. All right, now this is where we're going to end. And this part here, this hormone part, I've redone as a different set of notes because I like the way I've organized it. So I'm going to use those notes instead of these pages over the next few pages. We might come back and talk about one or two things on these pages, but for the most part, I'm going to go ahead and not use the rest of the notes and use the ones that I've made already. So take a look at those. Be ready for those in our next lecture, and I'll see you then.